computer. So if I share the screen, yeah, okay. So now why I'm not seeing, not share there. And so now I can do my PowerPoint. But this is not the PowerPoint that I need. Oh, hold on, guys. I got a bunch of PowerPoints open, turns out. Ah, here it is, okay. Close this thing. Open up. All right. So am I recording this? Not yet. Is it being recorded?
Liz, it looks like it's recording because I can see it.
Yeah,
too.
it has a little, yeah,
Yep.
okay, so good. All right, so thank you for coming. And I'm just gonna tell you about some new work in the lab that I've been leading. So just to give you a little bit of background, the lab started to develop this methodology to adapt the precision medicine platform to psychiatry. And in precision medicine, you have all these layers of network, patient-contributed data, clinical tests, behaviors, exposures, microbiome, genomics, and you put all that together and you deploy personalized, targeted treatments that are tailored to the person's genome, microbiome, and so on. And so the idea is that you take medicine from a one-size-fits-all model into a stratified model that kind of looks at subtypes of different phenotypes, and then eventually you make it into the precision medicine targeted treatment model. But in trying to adapt this to psychiatry, what I realized was behaviors was a very weak link there, because it leaves a gap between all these layers down here and the more refined level of inquiry at the molecular and the genomics level. And so we started to work towards that and took advantage of the biosensors revolution, which happens just a few years ago, really. And to that end, we defined different movement classes because movements make up all the behaviors that we perform at a macro level of description that we observe, but also at a micro level that escapes the naked eye. And instrumentation can tell you a lot about that micro level. But to that end, we started to develop a phylogenetic orderly taxonomy of the nervous system and realized that we could connect that way to the genomics. So I'll tell you how that could be. It's a proposition, of course. And we developed this intelligent behavior analysis paradigm where we combine and augment methods that rely primarily on observation, which is what we can see and we can unambiguously detect, with methods that rely on a finer layer of granularity of our behaviors and capture a lot of aspects of it that we're not missing in the diagnostics that we have. And more importantly, we started out about 20 years ago doing this with high-grade instrumentation from the lab, but then realized with the wearable sensors revolution, we all wear these things, and we have instrumentation that off the shelf is affordable and can then transform the way in which we do behavioral research. So we started to develop methods for Parkinson's disease and several neurological disorders. So here's an example of for example, the center of mass. And the idea was to first stratify automatically a random sample of the population. So a cohort of people come to your lab and you want to stratify them. Here's an example of a signature in Parkinson. These are control people. I'm just gonna focus on the first column here. These are control people in blue. This is the center of mass, the trajectory of the center of mass in 3 dimensions. This is a person with Parkinson's disease in very early stage. And this is the universal Parkinson's disease rating scale of 16. And it increases as the severity goes up. And as you see, the severity goes up, and there's like a disintegration of the center of mass trajectories. And so this is a very easy way to stratify based on severity, and in agreement with a scale that exists already in neurology to detect and to track Parkinson's disease. And then we added other conditions with a metronome, and we asked people to breathe at the pace of the metronome and so on, to see how these patterns change and so on. Because in the change also we can detect other ways to stratify the cohort. In this case, this particular parameter, the center of mass of the gait, gives it away very easily. So you can measure the departure from the neurotypical case very easily, but in other cases, it's not as obvious. And so the raw data that you have then has to undergo a number of transformations, and eventually you get nonlinear transformations, and eventually you can stratify digital data that way. So here's an example of that. We created a data type called micro movement spikes. And the idea was to make it compatible with cortical spikes and begin to also connect with that layer of data, cortical spikes, and this would be peripheral spikes. So to connect the central nervous system's spikes in this way. And the spikes are the peaks of any waveform. For example, here's the speed in meters per second contains peaks and valleys. And so we go through the peaks and valleys and we measure the fluctuations, and we scale that in a way that accounts for anatomical differences. And we can then put everybody on the same kind of parameter space. And in this case, instead of averaging the peaks under some assumed distribution, we use maximum likelihood estimation, and we get the family of distribution that best fits the histogram of those spikes. And then we plot them in a parameter space. And through empirical estimation of thousands of individuals, we now know empirically that this region in green here is a good region to be. This is a parameter space that plots each point here as a location based on the coordinates would be the shape of the distribution and the scale of the distribution. And the scale is actually the noise to signal ratio in the case of the gamma family, which fits well in a maximum likelihood estimation sense, the behavioral spikes that we get. And so the gamma turns out to be also representative of a gamma process, which is actually part of the Poisson random process. So it fits well, and we'll see later in another example, with a kind of model that we've been using in computational neuroscience to characterize the spikes trains. And so in the red here is where you don't want to be. This is high noise and very exponential distribution, very random. So unpredictable. You want to be here where the noise to signal ratio is low and the predictability is higher on average. So that's kind of the overall method. But then we can also, so this is an example of the data from the Gait that I just showed you, how to stratify controls versus Parkinson's in each one of the cases on the gamma parameter space. But we can also take compute the moments. There are formula to compute the moments of the gamma based on the shape and the scale. So we can then augment with other dimensions. And sometimes this projection will not tell you a stratification as much as this projection, as this lifting to another dimension will. And so it's what we found over the years is that sometimes the raw data won't tell you. Then the first transformation will stratify the cohort, and maybe there is another index that you can come up with. And because this data is continuously tracked, you can come up with these sequences and so build ways to stratify. This is another example. So I just walk you through some examples of children with SHANK3 deletion syndrome that go on to receive a diagnosis of autism. And this was a 2.5 clinical trial that we ran for a group in Mount Sinai. Joseph Buxbaum and Alex Collefson ran that group, and this was a trial on insulin growth hormone factor. And these children are very cognitively impaired, and so the only thing that really we could do to measure was to measure gait. So they came to the lab, and we built a platform that they would walk, pace back and forth, and we wired them 15 sensors so we could see the entire body in motion. And using that information, we plot them on this shape scale parameter and characterize their fluctuations in various gait parameters, in various, in this case, acceleration, angular velocity, this angular acceleration in this case. And here the open symbols are when they started, baseline with no drug, and then this is when the drug, after the drug. So we take these measurements and it captures the change due to the drug and the differentiation between young kids up to 8 years old and older kids, adolescents. It's a very small cohort. These were 16 participants, and then 9 of them were tracked through the 2 years. The others dropped, either just they didn't continue to come. These were from all over the US, by the way. They would fly into the New Jersey, stay overnight, come to our lab, and then go to Mount Sinai to do the rest of the testing. And then we always compare them to neurotypical controls of their age, and to autistic, idiopathic autism, and to ideal cases like a professional athlete and so on. So this is the scale goes up to 9, so they would fall in this, and the change, they will start very exponential and fall or very skewed, and then shift the distribution with the drug. So this is capturable by this micro movements paradigm. And so this is another example. It's a simple pointing task that we carry on in the lab, 60 participants of different ages and different levels of verbal skills. And here's the typical neurotypical control, and we're plotting these rastergrams much like we would do with cortical spikes. So each row here is a trial, and this kind of beige or orange, I don't know the color, spikes are from resting state, and then there's the movement onset, and then there is an acceleration period to the peak velocity, and this is a ballistic phase, and then there is another ballistic phase towards the touch. These trials are aligned to the touch, and then there is the retraction phase to rest, and then the offset of the movement. This is tremor again. So this is a typical person, neurotypical control. It's very periodic. You can see that there is, the periodicity of this. As we go up in the autism spectrum, this is a case of a Asperger's, the old definition of high functional Asperger's. So you still see some structure, but now you start seeing more noise in the ballistic phase, which shouldn't be there. And you could think of it as some type of tremor. Then you have a sort of a child with some verbal ability, not too much again. But by the time that you get to minimally verbal children, this is a typical case. You have a lot of noise, a lot of spikes where they shouldn't be. And so we can then group them in a frequency histogram, and what we see is the typical control here in the bottom here has a tail, which is essentially these are the high speed peaks. This is the mode of the high speed, and this exponential is the tremor peaks. So if I go back, so the higher values here of the peaks fold over here. So this is amplifying it, so you can see that it has a distribution with the mode. As you go up all the way to the non-verbal, the minimally verbal, you see that that is smaller and smaller, and we built a ratio that measures the exchange, the interchange between those two modes. And so the higher the ratio, the more turns out the higher the verbal ability that we found. And then we model it. This was work by Dee Wu, who's now at Apple, one of our students. And she was a theoretical physicist, actually. So she modeled this as a Poisson process. This is totally random, semi-periodic, and periodic. And so this would be the case of the typical control, and it would fall over here. So this gave us a way to measure this behavior and to stratify autism in terms of age and separate them from controls. And then another thing that we've been doing over the years is quantify all this digital data in relation to the inventories that exist already in the clinical domain to see how congruent they are. And they were a good indicator of IQ levels. So the higher the rate and the higher the verbal abilities, the higher the IQ. Also in the ADOS severity, so the higher the severity, the lower the rate parameter. And so it was congruent with several of the inventories. This is another case where we did the same with involuntary resting state motions and also more with decision making. And autism really always falls out there in a different domain of the noise, the variability. We also digitized a lot of the cognitive tests. So this is the Montreal cognitive assessment test that people do in neurology, and we can also stratify people based on their performance during the test, just digitizing their drawing. So this is the drawing of a clock at some particular time of a control and a person with Parkinson. And we also digitize the faces when they're taking this inventory, so we can tell which area of the face is more engaged and changing the most, and so on, when a person is like on a neutral or in a smiley face, and so on. Because in Parkinson and in autism as well, the dopaminergic system seems affected, and that has some relation with the muscle tone in the face. And so you get in Parkinson what is called the mask, the face, face mask effect, where the person shows no affect because the muscles have lost the tone. And in autism is very common as well, but it's not as recognized. So with that in mind, we translated all this high-grade instrumentation and essentially, put our lab in a box. And literally what we do is we take all these instruments that are now off the shelf. This is like $90. This is more expensive. It's like $299. This is like $100, top of the line webcam. And this is the most expensive item, which is about $1,500. This is an FDA approved sensor that takes muscle tone and heart respiration and heart rate and so on. And all of these we put on a box for about $50 to $60, including the insurance. We can then deploy that to the home. And with COVID-19, this is what we've been doing with protocols. And it's just like buying your Roku stick and running, the instructions. The instructions are visual, and so people can collect data at home. And so these things go to the cloud directly, and these ones that have to be saved, we have made protocols to get these in like a button press, and the person then saves the data. So they are designed to probe different levels of the nervous system control, voluntary, involuntary, autonomic. And just essentially we can profile the whole functioning, and even with the pandemic, we can do this. So this is something new in the lab, but it's important because we can lend continuity to behavioral research, which was disrupted for a while there for a lot of people. Now, what we realized was that there is a way to connect to genomics through the 3 fundamental types of muscles that make up everything in our nervous system. The cardiac muscle, innervating the heart and having to do with respiration and this autonomic control. The involuntary, the smooth muscles, we control all involuntary, they line up all the organs and the involuntary aspects of our behaviors. And the skeletal muscle, which controls all the voluntary and automatic type of behaviors that we learn, goal directed and so on. And we wanted to then change sort of from a exclusively top-down approach to a bottom-up and understand the peripheral nervous system influences on mental health. And so what is it that we can do with off-the-shelf technology and with, you know, more of a holistic approach to help support the nervous system and to redefine these mental illnesses as you know, of neurological disorders that have, for which there is accommodation and support out there in the neurological world. So with that in mind, we set up to look at the gene expression of these tissues, on the various tissues. And so that's the talk I'm gonna give today. So in the precision medicine paradigm, what we have incorporated is the digital behaviors that I just told you about. It's a way to stratify disorders that are heterogeneous and on a spectrum. And then the tissue is what we're gonna study, the gene expression and ways to characterize different disorders. And so that's why I call this precision medicine for psychiatry. And again, this is like textbook information here that we're all familiar with, but it's in a field like autism, it's completely unknown. It is not, doesn't enter in any of the curricula or certification. So we have very precise maps of the trigeminal nerve in the face that have to do with all the social axes, you know, eye movement, mouth movement, hearing, reception of a speech and production of speech, all the muscles that innervate for speech production, and the smooth muscles that are necessary for swallowing and for speech, and share the speech apparatus as well. And then the dorsal root ganglia, all maps that we have throughout the body, and so on and so on. So in a way, ignoring this for so long in autism has been, it's really a tragedy, because these are all the things that we see that are wrong with the kids from the get-go. And, you know, pain, temperature, tics, dystonia, light, chorea, food aversion, which comes also from the smooth muscle and skeletal muscle problems, dysautonomia, food aspiration, which leads to pneumonia, and then body coordination issues like ataxia, apraxias, and so on. So this all leads to a very rapidly and accelerated aging that by 20% of the population, autistic adults, has Parkinsonism signs. And this is in contrast to typically aging adults, which only 0.9% after 65 have that. So it's really tragic that we don't pay attention to this from an early age. And I have done a study of this systematically with aging, and by the age of 40, indeed, there is a departure, a fundamental departure. The blue here is a trajectory across all these. This is thousands of individuals in open access database. And this is a large departure. And that's about the time when you start seeing the Parkinsonism very clearly. So things don't add up, and so that's sort of the motivation for my research program, and trying to connect the genomics through the tissues, and then the tissues to the neurology, and the neurology to the behavior, so that we we can kind of navigate these phenomena differently. So in things like autism, schizophrenia, depression, which are defined, mental illnesses defined behaviorally through the DSM and through other means, we have all these neurological disorders like symptomatology that anyone who studies the neurological disorders immediately sees them in these other mental illnesses. Like in the lab, we have investigated all these disorders, and at the level of digital biomarkers, and characterized the motor and sensory motor issues. So there is a behavioral pipeline of diagnosis and behavioral modification treatments, pretty much based on Skinner conditioning and type of deal. And so the burden is on the person. There's also this narrative that the person is uncoordinated, that it has OCD behaviors and rituals, that has no feelings, no empathy, and so on and so on. The burden is on the person, but the question that we pose is like, well, how about the underlying neurology? Can we do something about it? And it's interesting historically that autism in the 70s was initiated with a model, a neurological model proposed by a neurologist, Ralph Maurer, and Antonio Damasio, who's still around as a philosophy of neuroscience prominent person today. And they were competing with a model by Lovas, a behaviorist at the time, who translated the behavioral conditioning for kids. And this happened at UCLA. And that model took off, and is the prevalent one in autism. And so it was the neurological model was abandoned, and we inherited Lova's animal conditioning model translated to these humans, kids, and then a sort of a disjointed genetic approach that doesn't connect to the Lova's approach in any way. So we also have schizophrenia, which also historically started out in the pre-Freudian times as a motor-related description. So it was all described motorically. And so it also lost that flavor over the years, and it has no connection to the neurology. And in general, there's this circularity where we have the genetic data labeled according to the DSM criteria and other criteria without the connecting to the underlying neurology. So at the moment, there's no blind to really take a random draw the population and based on their behavioral phenotype, have an estimation of what underlying genetic causes may be in all these disorders. So, so it's really the question for me in the case of autism is like, why not go back to the neurological model? And there are many reasons for that. We have 2 decades of brain science that have been funded by the decade of the brain by President Bush program, and then the brain initiative by President Obama program. And we have all these conditions in autism that do have a neurological explanation. And we have other disciplines that provide accommodations for these neurological conditions and that help the person improve their quality of life. So among them are the ataxias, Parkinson's disease, mitochondrial disorders. And these are different communities that already have in place treatments that help the person live a better life. And what I want you to take out of my talk today is something that we may not understand the underlying causes of autism or all these disorders, but we don't have to understand them to be kind to these people and to actually have a model that accommodates their condition. And so that's sort of what I'm advocating, that we, instead of sidelining the neurology like the DSM or this Autism Diagnosis Observation Schedule do, which are the instruments to detect autism or to officially diagnose it. We should acknowledge the neurology underlying this behavioral phenomena, and then just see what they have in common with disorders that are officially accepted as neurological disorders. So my question in this talk is like, what do they have in common genetically speaking? What is something that can tie them to do we get gene expression in 54 tissues that are common in open access databases, pointing to a neurological issue such as motor, sensory, emotion, and memory in autism that is common to the well-known neurological disorders. So out of these 54 tissues that we get in open access databases, which one are the ones that are affected by eliminating the genes that are associated with each one of these disorders. And so we have autism, depression, and schizophrenia, and we have other non-neurological disorders that I will be looking at as a control, like different types of cancer and autoimmune disorders, diabetes, PTSD, which is acquired, and other like mitochondrial disease. So the idea is to see if we have mental illnesses that have gene expression in tissue that is common with well-known neurological disorders. That is, if we eliminate the genes that are associated with each one of these individually tested disorders that are rendered as mental illnesses by the DSM, do we get anything in common, any convergence with neurological disorders? And then how about non-neurological disorders? What do they have in common with well-known non-neurological disorders, and how do they diverge? So that's kind of the question. So what I did was interrogate the genomic information that is offered in the SFARI genes module, for example, which is open access, and also the GTEX portal, which is a portal that has information about all these 54 human tissues, and their gene expression is constantly updated. And so you can enter a gene and you can see how it's expressed in all these 54 tissues, what is the gene expression. We also have the online Mendelian inheritance in man, which you can enter a gene and then get all the diseases that this gene is associated with, and all the neurological, in the case of the neurological ones, you can even get sort of a list of accommodations or pointers to that. And then this is the opposite, that here you enter a disease and you can get comma separated value files in Excel formatted files where you have all the list of genes that are associated with that disease. And so you can also go there. And of course, you can also go to the literature, which I did. I did several literature reviews to extract genes that are present in Parkinson, in Ataxia, recessive and dominant, in X chromosome disorders. And the reason for that is because there is overlapping with the genes that are associated with autism. And so I wanted to see how that also has an effect on the gene expression of the tissues. And so in the SFARI autism scores, up to the day that I did the interrogation, there were 906 genes declared and grouped in these 4 modules according to the literature evidence. So they're ranked. And then there is all these genes that can reported in this genet, in that portal where you enter the gene, the disease, and it gives you the list of genes. And so we can then go and see also these major mental illnesses, depression, and also I also looked into unipolar and bipolar depression. And then I also did the search on the different types of cancers and diabetes and all other types of non-neurological disorders, and in the mitochondria, which are mixed. So the way that the gene expression is reported is you have a matrix of RNA sequence genes, about 56,000 plus, and by 54 tissues. So what I did was transpose that and express each one of the 54 tissues as a function of the genes, and then normalize the count by put it, so that way I put everybody on the similar standardized scale from 0 to 1. This is the normalization I came up with. These are the 54 tissues in alphabetical order, and this is an interrogation for one gene, the CHANK3, which I had studied in the clinical trial in the lab. So I was curious about all the brain tissues listed here in yellow. You can see that, for example, the cerebellar hemisphere and the brain cerebellar are the highest, they have the highest expression, and so on. So you can, for each gene, you can actually plot this in GTEX, and it will tell you what gene, what tissues are the most expressing that particular gene. So you can go to do the same now. This is a different type of representation, but of plotting, but it's the same alphabetically ordered tissues, right? And what we see is all the genes expressed here. And what I'm doing is coloring in red the autism genes of a score 1. That's the highest score, meaning the ones that are highest associated, the most evidence associated to autism. And you can see that, for example, cerebellum and frontal cortex, and you know, you can see that what are the ones that are highly expressed. And so the idea was like, what if we eliminate those genes and recompute the gene expression of the tissue? What tissues would be the most affected? And so this is another way to plot it in a color map, where you have the color is the transcript per millions and log transcript per millions, and then the color indicates that. And so blue, there's not much here, there's a lot here, and so on. You can go back to see what tissues are the most expressed. You can do it for different types of disorders to see how they interact and so on. And so this is like kind of the methods in the paper, if you had a chance to look at it. I take the log transcript per millions count, and then I normalize it by that formula. And then, so this is the histogram of these. So this is, for example, these are for four different tissues. For example, the brain frontal cortex, the brain frontal cortex, and the different genes, and then different. So the SFARI. SFARI stands for Simon's Foundation Autism Research Initiative, and that's the score 1, the score 2, and the score 3, and the score 4, which are the syndromic genes. So these are an example of autism genes in the SFARI, and then one tissue, and then how they change in that tissue. And this is a different plot with the normalized count in different tissues, like the basal ganglia, the heart ventricle, the frontal cortex, and the musculoskeletal, and then how they change after the normalization. And so this is the distribution that we come up with. Is an exponential distribution. So that led me to estimate the rate parameter, which I explained in the paper, how to get that lambda estimation. And so once I get the lambda for the original gene expression, and then for the gene expression on the tissue after eliminating the genes associated with the disease. And I can then measure the change. So the question that we ask is, given the known neurological phenotypes of known disorders like Parkinson's disease, the Ataxias, the X chromosome, the Fragile X, and the mitochondria, is there convergence between the most affected tissues for psychiatric disorders that are defined by the DSM as measured by this stochastic change? And the stochastic change comes from, again, computing the lambda parameter in the exponential distribution, characterizing this stochastic process, which is the sequencing, the gene expression. And so estimating through maximum likelihood estimation, and then through a number of steps in the paper, this is the value of lambda. So it's the number of sequence divided by the sum of that. And so given the series of genes transcript per million counts per tissue, we can obtain a characterization of the gene expression and visualize it using our methods, and the reason for that is because the exponential distribution is a particular case of the gamma distribution. And so we can sort of recycle repurpose the methods that we had shown in the beginning with the gamma moments parameter space and utilize that to visualize what we're doing. So this is sort of what I do here. This is the computation for the schizophrenia gene. So I eliminate the schizophrenia genes just to give you an example. And then I recompute the gene expression. And then this is the subtraction of the gene expression with all the gene and the gene expression without the schizophrenia genes. Then I median rank that into 4 parts, and this is the highest median ranked group, and the highest, the 2 most affected tissues here are the amygdala and the basal ganglia. So then I expand here and show you this out of the 54, the first 13 most affected tissues. So the basal ganglia, the amygdala, the basal ganglia, the heart left ventricle, hippocampus, substantia nigra, and so on. And what I started to notice immediately is that in all these, and we will see that in a moment, in all these mental illnesses, performing this exercise, what it reveals is that all these subcortical areas related to motor control, memory, and emotion and regulation, also the hypothalamus shows up a lot, are affected. So this is a plot of the gamma moments, the mean, the variance, the skewness, and the kurtosis in the size of the proportion to the size of the marker for all 54 tissues. And the open circles are the GTEX full genome, and the colored circles are after the removal of the genes associated to schizophrenia. And this is the lambda delta, the difference in the color. And here you see the first 13 tissues in the top ranked, median ranked group. So you can see that there's a shift, a stochastic shift, and quantifiable. And this is also how you can see it in the exponential distribution, which sorts out the by value. So this is sorted out by the values, and you see the difference as well. So all genes in black and removed in red. And so we can do this now for each one of these disorders and see what kind of convergence we get. This is the autism in the SFARI repository, and this is autism in the DisGenet repository. And we get that in all cases, again, we get brain, substantia nigra, basal ganglia, hippocampus, amygdala. So these are the top tissues that are affected. In the ataxias and Fragile X, which are well-known neurological conditions, we have also brain, but we also have heart affected issues. So this is also very informative. This is another way. I use something called the air movers distance, which computes the difference between 2 frequency histograms. And it's sort of a measure of the amount of effort that it takes to compute, to change one different histogram into another. So computing the 2 histograms of the lambda, according to the lambda, and according to the gene expression, then we can see the different tissues as well. And there's congruence with the other method, which is just the absolute difference between the 2 lambdas. That's for autism, ataxias, ataxias, and then I go to the DSM mental illnesses, schizophrenia, depression, unipolar depression, bipolar depression. And again, you see a prevalence of these subcortical structures and the heart as well in the depression case. And the musculoskeletal also shows up a lot. We will see this in a tabulated, color-coded in a moment. The different ataxias, you can see the Gait ataxia, spinocerebellar ataxia, cerebellar ataxia, progressive cerebellar ataxia, and so on. And again, the ataxias seem to have an influence of a manifestation of the heart ventricle as well. So this is in tabulated form. So all the blue, the dark blue are subcortical brain structures. Then I color in red the heart, and then in light blue the muscle skeletal, and then any other tissue that has like muscle, a smooth muscle, or related to other functioning are in brown. And so what you can see is that in autism we have a lot of the subcortical structures that are expressing changing the gene expression is affected by the removal of the genes. And a lot of them are congruent with the neurological conditions. And then I also eliminated those ataxia genes and Parkinson's disease genes from the SFARI and recomputed to see if the reshuffling would change the structures, but it remained as subcortical, motor control, emotion, and memory. And so what else? So something noticeable is that the heart is present in a lot of these disorders. And also there is musculoskeletal. And then when we get into the depressions, there is also so how about non-neurological disorders, right? What do we get there? So there we have other organs, and only at the end of the first ranked, median ranked, we start getting some of the brain structures. So the manifestation, as you could imagine, is different. So we get a lot of heart as well, and pancreas and liver and so on. Colon cancer, we don't get any brain whatsoever. It's all pancreas, whole blood, and so on. Breast cancer. So I'll present this in a tabulated form. It's easier. The mitochondria is a mix of musculoskeletal, half ventricle, and other organs, and the brain subcortical structures. There are different types of mitochondrial diseases, and they overlap a lot with autism. So a lot of these kids go on to get a diagnosis of autism as well. And then there's PTSD, which is acquired and is kind of a mix between brain and non-brain tissues. So here's the tabulated, color-coded for non-neurological disorders. And what you can see is that some of them, like particularly diabetes, is mixed. So there's a lot of, in diabetes, affected tissues from the removal of those genes that in the second half of the rank are more brain related. But aside from that, you see that in the first half is primarily non-neurological and heart and muscle related. This is another way to present the data. So this is a color map where the color represents the delta lambda affection by the removal of the corresponding genes to that disease. And then here you have the tissues that are mostly affected. And so these are the mental illnesses, autism, schizophrenia, the depressions. These are the neurological disorders, and these are the non-neurological ones. And a way to actually look at it, thresholding it to see what we get, what patterns come up is this. And interestingly, so there is a difference between the mental illnesses and the neurological disorder. In the whole blood is does not affected when you remove those genes in the mental illnesses, but it is affected in the neurological conditions. There's also the big difference here is that the non-neurological conditions barely have any subcortical structures for motor control, memory, and motion regulation. And then we have musculoskeletal is present across a broad range, as well as the heart ventricles and so on. So that's about it. I, from this, conclude that removing the genes associated with mental illnesses in autism, schizophrenia, and depressions alters gene expression maximally in tissues related to motor control, motor learning, adaptation, and coordination, followed by tissues related to emotional memory. There is overlap between maximally affected tissues in well-known neurological disorders like the Ataxias and Parkinson's disease, and those maximally affected by removing genes associated with mental illness from the DSM. So motor, emotional, memory issues can be all neurologically addressed along with accommodations that we have from these other fields to help patients to cope and flourish in life. So the question that I pose is, why not add neurology to those definitions? So this is sort of the conclusion from this first pass to this method of eliminating the genes, but it's very crude way to do things. So the ideal is also a static way to do things. So it's just a snapshot of what you may have. So a better way is to actually do it dynamically over time, and in the context of, for example, growing cell lines with induced pluripotent stem cells research, and that's something I'm going into. And also, instead of removing the genes, to actually play with the gene expression with several functions that could tell us different scenarios, like overexpressing them or turning them on and off, and so on. And so to that end, the future directions, I'm collaborating with the Salk Institute on this. We have been curating data samples of the transcriptome that have all the genes. And one of the problems that we find is that in the field, there is a tendency to oversimplify, because there's so many, the space is so huge, you know, you have 56,000 plus dimensions there to deal with, turning on and off with different dynamics. So what people tend to do is some form of fancy PCA and eliminate a lot of those dimensions. So you work from, say, 26,000 to 5,000 genes, and then you track those in as the neurons as the cells become neurons, or neuron precursor neurons, and so on. And so what I did during my sabbatical was create a new dynamic interrogation of the stochastic transcriptome, meaning the trajectory, meaning as dynamically as they change. So I won't get into the details because we ran out of time already, I think. But essentially, it's really cool because what they do is they grow the baby neurons from the same person. The same person, they grow neurons in the embryonic stage, and they also grow neurons, age equivalent neurons. And so you can kind of, yeah, the way I see it is like time travel almost with the person, and see disease from a completely different perspective, as how these neurons develop, the cell lines develop, and you can apply all these stochastic methods. And so I did that with Simon Schaffer. Let me just skip through this because it's a different talk. And we interrogated the transcriptome, over 26,000 genes there, and in a paper that was published. And again, this they do PCA and project everything to a space, and then these are the different days, the 12, day 0, 12, 19, 26, 40, 54, and they follow the cell lines until they become neurons, and then the idea is to classify the neurons. But by the time you get to the neuron stage, you have already eliminated all these genes. So what I did is not eliminate the genes. And so I'm going to skip through that, but I just want to show you the little, so the methods are similar to what I just explained, and I just end up with a particular stochastic trajectory for each gene. But here's how, this is the transcriptome, and this is a color code based on their cumulative distance in probability space that they travel, and you can see how they change over time. And so without losing any information, you can actually track the gene expression. And at this point, they have neurons. So those neurons can be classified based on the full genomic fate. This is the fate of the genome, of the transcriptome, and the state, which is the underlying changes in a state. So these are or some are turning off, some are turning off, and so on and so on. So that's a new line of research in the lab. It's more of a computational line of research, and that's what we're. So what we plan to do next is combine this interrogation of the disease to eliminate the genes across these dynamic trajectories and see how the cell classes shift around. I think I'll stop there and take questions. I think I have to stop sharing, right? Sharing. Stop sharing. All right. So that's sort of what I have, and I'll take questions.
So Liz, I have a question. So that's an amazing amount of beautiful data. And as someone who also thinks about studying gene expression, and I do it kind of one hit at a time, I'm kind of in awe of what you're doing. So I have a couple of questions. One question is kind of more broad, which is what's the plan, or how do you envision, in the next 10 years bringing this back to the clinic? Is the idea that then you kind of know which genes to screen for, and this is like a diagnostic kind of effort, right? To I don't know, somehow modify the genes in utero and then you reduce the autism or whatever, the psychiatric disorder, or is this something beyond that?
So for the clinic, so the idea here was to characterize it first, because to take it to the clinic, you really need to have a validation, like a massive. So the first thing is, there are all these open access databases now that have all this genetic information. So the data is already there. But what we don't have is a way to utilize it such that you can interrogate it automatically and say, given this phenotype, what is the likelihood of certain genes being affected, right? So we don't have that. And that's what I'm trying to do massively with off-the-shelf technology now that you can send home the person with, and
I love that. You told me about that last week. I think that's fantastic. You can just ship it in a box.
Yeah, just ship it and give me back your digital biomarkers, and I'll put you in a map, and then I know that you fall here, and this is where the other group falls, and so you automatically stratify the population. But now the tissue is sort of important, because neurology already has cataloged a lot of these issues, right? Okay, there's gait issues, there is memory issues, and neuroscience has sort of a way to connect that information with the brain. So that's where the tissues come in handy. That neuroscience already has kind of a map from brain to disease. So we can then make that connection and go back to these databases. And basically my vision is to train neural networks to do this and inform, like, give me, this is the phenotype, give me the likelihood of the genes that are going to be implicated. And then we go back and so on. And it has to be refined, obviously, it's very crude right now. But
yeah, okay, so the idea is something like, once you have that true kind of fingerprint of what that particular person has, both, I guess, biologically and behaviorally, then that steers you in the right direction for precision medicine to really make a real difference. I see.
Right, because that way, we're repurposing information that's already there. Of course, we have to refine this, right? And so, AI is still very brittle. AI is very fragile at the moment. We can't rely for a diagnostic on that alone. You have to combine all these sources. But eventually, if we do this right and massively phenotype people with this off-the-shelf stuff, which every day becomes better in terms of resolution. You know, 20 years ago, I could only do this in the lab with sensors that sample at 200 hertz. And today you wear a Fitbit that samples at that much, you know? So you're like, yeah.
Yeah, that's amazing.
Yeah, and so for example, OpenPose, which is a very good software, free for academic research purposes from Carnegie Mellon, we've been using that a lot in the lab to do facial expression, sentiment analysis, and a skeletal analysis and hands analysis. And so you do that with a webcam. All you have to do is be very consistent about the distance and the angle that you place the camera. So we buy a tripod that comes with instructions to the person precisely where, how many centimeters from the camera that you have in front of you and what angle and so on, so that we get the exact same data from everybody in terms of shadow and, you know, depth and whatnot, which influence all these video-based skeletal kinematic kind of deal. So through a lot of effort and you know, my students have been simulating this in COVID times. So they send this to themselves, like they simulate the entire process. From the moment they have to pack everything in a box, send it to the post office delivers it, the other student gets it at home, has to clean it. And so when we give a protocol to the person, we give it everything like it's in real life.
Yeah, yeah.
And IRB approved. All of this has to go through IRB.
That's right.
So it's like simulating COVID times. It's just that somehow we didn't know COVID was going to come, but we had already worked on. Yeah, it's like just serendipitous. We had worked out this whole thing because we were working with populations that sometimes couldn't make it to the lab, you know, and we had to do this at home and so on. Like, so my students were already in Parkinson's evaluating the emotional expression because of the face mask condition that exists in Parkinson. We were evaluating that remotely already. G was doing this already and things like that. So it's repurposing really what we've been doing for a number of years and adapting it to the current situation so that we can lend continuity to behavioral science and to clinical behavior, because a lot of the people in autism are out of the job right now as we speak because they can't do their testing in person. So here we have a way to do it remotely, just the way they used to do it, but now over the internet. So.
Yeah, that's fantastic. Yeah, so my second question is a little bit different. I'm thinking about all these different genes and different populations of genes that are changing in their expression. I love the dynamic experiments. I think that's going to be just a gold mine of information. It's really exciting. So in that kind of context, you know, as someone who thinks about epigenetic mechanisms, I wonder if there's a way to use the same technology that you're developing in a different way, which is to use kind of the data to identify which genes seem to be co-regulated. And the idea would be that maybe co-regulated genes are regulated by a particular epigenetic modifier, right? And that could kind of create these families or subclasses of genetic changes that you could then use to predict what might happen in, you know, other people, but then it would link it back to a particular epigenetic mechanism. I think that's particularly important and maybe useful for data mining in these populations that you're studying, because a lot of the psychiatric conditions have a kind of experience component, like an epigenetic component to it, right, as you go across the lifetime. So I don't know. I don't know if that's a question or just kind of a comment, but it seems like there's something that could be done there just to think about the genes being co-regulated and differentially expressed together. Maybe that could link that back to you know, a single mechanism.
Yeah. So in the transcriptome work that I did back at the SALC, the way we handled the genes co-regulation was through network analysis. But I think that it needs And so that automatically separated certain genes expressed at certain times, because, so to put it in context, the original traditional way to do it is just to eliminate the gene because it's not there, say, on day 12. But that same gene appears on day 19, turns on, and so on. So what is that gene doing to its neighbors when it turns on, right? And so in that way, by not eliminating the genes and tracking their on and off states, we handle that. But what we don't have in there is, for example, I think there are two important axes there, which are metabolomics and the microbiome. So those two are a mystery to me because obviously this is not my field. But I'm starting to see collaborative work with people in the microbiome world and people in the metabolomics world. We're running a small clinical trial right now through the biomedical grant. I mean, we're about to start. COVID kind of stopped, and then we normalized with the box, the lab in the box deal. And so now we're starting. But essentially, we want to use these three axes, right? One is the nervous system, the other one is the metabolomics, and the other one is the microbiome, to understand how they interact. And I envision that for genomics, that would be the way to do it. How are these two axes impacting this other one? I mean, the nervous system is just the low-hanging fruit, because with the wearable sensor revolution is what we can readily and cheaply do, but clearly it's a systemic problem. So you'd really need to look at the microbiome. And frankly, I think the microbiome is the future of medicine. We just don't understand it yet, but yeah, that's sort of my take on that. But I mean, it's just very all exploratory. So, any other questions? People feel free to, don't be afraid of asking. You know, no question is, oh, you know, worst case scenarios, I don't have a clue, and I'll just tell you that.
Liz, there's a couple questions on chat.
Oh, the chat. Okay, I forgot this technology. How homogeneous are the tissues, age, sex, collection date, Mimi? So Mimi, I don't know that information. I can tell you age. I mean, the age and sex you can get from the database. In this case, I didn't break them down into sex and age, but that's a great question. And perhaps I could get at the issue of the dynamics by breaking it down by age. So some of the databases do list the tissues at different ages, and that is exactly what Simon and I are doing right now. Yeah, that's a very good question. And the answer is that the data I analyze here, it's all bundled. So it's not broken down into age, but there are databases that have it broken down by age. So that's the next step. So with these methods that I presented, then I will be interrogating by different ages. So they have like induced pluripotent stem cell lines out there, and they develop all the tissue gene expression as a function of age. Not an age of the person, but age of the cell line developing. So you can do a dynamic interrogation, which is the next step. Then John, I have to leave at 1:15 call, but I just wanted to say that it's lovely talk. Okay, great. That's always really thanks. Mimi, fun. Thanks. Will you be able to collect tissue from your lab in a box subjects? No. That would be, you know, to collaborate with the Salk, for example, we had to pass the course, get some kind of, for biospecimen. It's so complicated with COVID, we can't. I mean, we do have, Rutgers has like a very good pipeline to get all this information and get like certified, so to speak, so that you can do it with this post office. So the post office, FedEx has the proper labels for the specimen that you're, so on. So we have a collaboration where we want to collect skin cells and ship it to the Salk, and then they will grow the cell lines for Fragile X families. But with COVID, we kind of step back and are thinking how to do that. But yeah, I wish. It's just COVID really changed everything. So we took the digital biomarker part and say, okay, we're gonna take care of this. The other part we're still thinking, because I mean, Rutgers has the mechanism in place for biospecimen and all that stuff. But it's new to us, so it's kind of a new enterprise. So we're kind of taking it easy. First, we want to pilot our lab in the box with the stuff that we're familiar with, and then we'll go to the genomic stuff, to the skin cells and blood samples and saliva and so on. But yeah, that's a very good question. All right, I think, anybody else? No? Okay.
Well, thank you, Liz. Really
Okay,
fascinating stuff as ever.
Thank you for coming.
Take
Bye.
care. Nice to see you, Donna.
Good to see you all.
Bye.
Bye, Donna.
Bye.